Today we're going to deal with the second chapter in the final section on water and salt balance, and today we're going to be looking at the in detail at the salt and water balance, first in freshwater animals, then animals that are ocean or saltwater animals, and then animals that may go between the two, and how they adapt to changes in salinity. And then we're going to look at terrestrial animals, animals on land, and the very specific problems that they have, where their problems are more in terms of water and volume balance rather than salt, although salt enters into it, of course. So let's begin by looking at the freshwater animals. Okay. Just some general points to begin with. Animals that live in fresh water now all descended from ocean-living ancestors. In fact, all major phyla of animals originated in the ocean. And a few other points here. Marine fish, fish that live in the ocean, have a lower osmotic pressure and ion concentrations in their blood than seawater. So they tend to lose water and gain ions. Since they're lower osmotic pressure, water tends to leave into the ocean, and their ionic concentrations are less than seawater, and so ions tend to enter in. So these passive diffusion challenges have to be met, controlled by energy-demanding processes. Freshwater fish, on the other hand, have a higher osmolarity and ionic concentrations in their blood than the environment. So they have the opposite effect, the opposite challenge. They tend to gain water, and they tend to lose ions. And again, energy has to be expended to restore this balance. And as it turns out, the blood of freshwater fish has lower osmolarity than the blood of marine fish. As part of the adaptation to freshwater, there's a decrease in the blood osmolarity. Now, so here we have a repeat of the table from the last lecture, table 27.1, just to remind you that the concentrations of the ions in seawater are, for sodium, 470 and chloride 548. So these are very high compared to any blood levels. And the values in freshwater are very low compared to any blood levels. And the other ions are listed here. And remembering that the salinity of the ocean is approximately 35 grams per kilogram of water, whereas in freshwater it's less than 0.5 grams per kilo. So keep these in mind now as we go forward and look at the animals in freshwater adaptation. Okay. All freshwater animals are hyperosmotic regulators. Remember, their blood levels for osmotic pressure and for ions are above that of the seawater. And here we have, again, some examples of the animals in this situation, where you have the freshwater mussel, osmotic pressure here is 44, and the ion concentration for sodium and potassium are very low. And then you compare that, say, to the brown trout. where we have osmotic pressure more in the range that we're used to seeing for mammals and invertebrates, around 300, and also with the values for potassium and for sodium and potassium that we're more familiar with. And then for the frog. And then for comparison, the numbers for river water here. So freshwater animals have a lot more osmotic pressure than their environment water, and a lot higher ions than their environmental water. Evolution of more dilute blood in the freshwater fish is an adaptation to reduce energy costs. So I had just told you that the osmotic pressure of freshwater fish is less than the osmotic pressure of marine animals. And so it appears that evolving a more dilute blood, or allowing a more dilute blood in freshwater fish, means that the organism has to expend less energy to maintain those gradients. Now let's look at the water and salt relations in a freshwater animal, and here we have a crayfish of some kind, and the points to be made here are that the blood osmolarity is around 400 milliosmoles with about 200 millimolar sodium, whereas the ambient water that is 3 milliosmoles and 0.4 of sodium. So you have the situation where the animal has evolved a impermeable shell, and so the main site of gain of water and loss of salt is from the gills. The carapace is not permeable. The gills, which are protected, they're underneath the carapace. You can't really see them from the outside. This is the site of the loss of ions to the lower concentration extracellular or external environment, and also the site of gain of water from the external environment. There's also loss of water through urine, and the urine is produced in these antennal glands, also called the green gland, and this is where the urine is produced in these types of animals. All right? So, the summary of all the changes now are shown in this slide. The upper portion of this, again, is we just saw this. And now we want to say that the principles here, that the passive exchange of water and ions will depend upon the gradient. So we already know it's a large gradient for both osmotic pressure and for ions, and also the permeability. And we said the carapace is not permeable, so that's okay, but the gills are very permeable. And then the surface area is the third. We can now generalize the idea that water permeability of the integument of freshwater animals is all very low. That's an adaptation that their skin or covering, the outer covering, which is the integument, has very, very low permeability to water. And so therefore almost all the exchange is from the gills. Now what's interesting about that, of course, is that the gills' surface area is needed and enhanced for oxygen uptake. So the more energetic an animal is, the more their basic metabolic rate is, the higher their oxygen consumption is, the larger the surface area for the gills will be. But that also means there'll be a larger challenge for salt and water flux, which costs energy as well. So the enhancing of the oxygen uptake aspect of the gills also leads to more energetic costs through loss of ions and water. So a balance there has to occur. Now, the thing about hyperosmotic regulators is that they have a high urine flow. So you have and so here's some rates of urine production in snails, crayfish, mosquitoes, frogs, toads, and goldfish. And you can see that their rates of water production here, 36 to 131 milliliters per 100 gram of body weight per day. So these have been normalized to a 100-gram organism. Also, when you do this, by the way, when you use 100 grams as your reference point, then the numbers also can be referred to in terms of percent of body size. So these snails are turning over, urinating a volume equivalent to 1/3 to 1.3 of their body weight per day. It's a huge water flux. Their osmotic ratios are all way under. The UP ratios are all very low. This means that in freshwater fish, that the kidney is the organ of volume regulation. Controlling this water flux is the way that freshwater fish, freshwater animals control their water volume. And just a reminder from last lecture, the UP ratio less than 1 in these hyperosmotic regulators, that leads to increased plasma osmoles and increased ionic content, but loss of ions is proportional to the urine flow. So volume regulation and ionic regulation are in conflict in freshwater fish. They have to increase plasma flow to get rid of the water, but that means they're going to have to lose, no matter how dilute the urine is, they have to lose ions by that method. So, how do they handle the ions? So, this figure that we have here, we've seen before again. We saw it in Chapter 5 earlier in the course. And here is the standard transporters, basically a active sodium hydrogen exchange pump, and chloride and bicarbonate exchange active transport. Both of these require ATP. The net effect of these pumps in the gills, in the gill epithelial cells, is sodium and chloride enter, and the source of the hydrogen and the bicarb comes from metabolism and body water. And again, there's a enhanced explanation of this figure if you go back to box extension 5.2. And the important summarizing point here, just as we said, the kidney was the organ for volume regulation. In freshwater fish, the gill is the ion regulatory organ. Now, in all these animals, the common features of the mechanisms that they have for active sodium and chloride uptake is that it requires ATP, and the pumps for sodium and chloride are different and independent. So that is what I just pointed out before. Sodium, for example, sodium can exchange with hydrogen in one pump, chloride can exchange with bicarb in another pump. And variations of of that theme occur in all these animals, with the point being that the sodium pump is separate from the chloride pump. Usually chloride exchanges with bicarb, and that keeps that exchange electroneutral. And sodium exchanges with hydrogen, and that keeps it electroneutral. So it does not change the electrochemical gradient, the electro part of the electrochemical gradient, electro part. Bicarb and hydrogen come from metabolism, and it's a way of getting out waste. And also, by slight changes in this balance, the system contributes to pH regulation. So these are the common features of the freshwater hyperosmotic regulator. Now, as I said, there's two functions of the gills, really, two major functions. One of them is related to oxygen uptake, and we've studied that already in previous parts of the course. And the other part we're looking at now, and that's salt and sodium and chloride uptake. Now, these functions are performed by the same gills, but different parts of the gill. So here we have a figure that shows that water flow goes across the secondary lamellae off the filaments off the gill arch filaments, secondary lamella. And the secondary lamella coming off of the gill filaments, these feather-like structures, have a huge surface area. It's a way of increasing surface area. Now, you remember from the first couple of lectures, in the larvae, the integument, the skin covering on the fish, performs both functions of ionic regulation, and oxygen uptake. You remember we showed a figure of the location of the sodium potassium ATPase going from the skin to the gills. The ionic part of the regulation shifts to the gills first at 2 weeks. So in the rainbow trout, as an example, 2 weeks after the hatching, the gills take on the role of ion transport, but oxygen doesn't switch from the skin to the gills until 3 to 4 weeks. And this is just another way of emphasizing that the two functions are separate and controlled separately, but occurring in the same essential organ. The slight differences, though, so just a slide that refers back to lecture 23, or the 23rd chapter, and you remember this is the way that oxygen is taken up by the gills. It reminds you of the structure in the opercular cavity, the flow pattern in the gills, and the water flow through the secondary lamella from the arterial end to the venous end. And that just reminds you that's how the oxygen is transported. Now, this is a good example of the gill where you have the filaments and the secondary lamella. And now this is just to show that from this same figure, where you have the ion transport aspect is localized at the base of these cells here along the base of the filament. These have been stained for sodium potassium ATPase, and there's some green ones here that are stained for the sodium potassium 2-chloride cotransporter. The blue is the nuclei of cells. So here the oxygen transfer function is essentially out here, but the ion transport function is down along here. Now the cells that are doing the ion transport are these mitochondria-rich cells, MRCs, mitochondria-rich cells. These are called chloride cells because they do a lot of chloride pumping. So these cells here in the center, this is the cell here. These are the ion pumping sites. They're surrounded by these pavement cells. These are the long, flatter blades, thin ones. These are responsible for the oxygen uptake. The chloride cells are responsible for ion pumping. And these mitochondria-rich cells, just to point out, that they can be either a freshwater type or a seawater type. Remember, the ion pumping is in opposite directions in the two cases, but it's still the function of the MRC. Now, if we look at the secondary lamella of trout living in ordinary fish water, fresh water, you see that these mitochondria-rich cells, these are the dark staining cells here. They've been stained for the NKCC transporter. And so you can see how they are distributed along the secondary lamella, and they're separated by these pavement cells. Now, if you take that animal, but you adapt it now to, and you put it in water that is of low calcium, what we call soft water, then the number and size of the MRCs is now increased. A lot more staining and a lot more increase. So these trouts are being kept either in traditional, regular fresh water or shifted to ultra low calcium salt water, fresh water. And there's an upregulation of the cells that are needed to get that calcium, what little there is, into the organism. Because remember, in the freshwater organisms, they all lose ions to their environment, and they need to have pumps that bring ions in. Calcium, of course, is a very important signaling molecule, and so in a low calcium environment requires increased amount of pumping sites to scavenge the small amounts of ions that are available. Okay, next we have a quantitative summary, in a way, of the freshwater water and salt relations. So again, we have our friend the crayfish, the same picture, but now we're putting some actual numbers on here. And here we have, we start here with this is a 29-gram crayfish, and it's at 20 degrees, 25 degrees C. Second. Yeah. 25 degrees C, that's right. And this organism, this animal, will lose 240 micromoles of sodium per day by diffusion out of the gills. And it will gain water by osmosis of 2.4 milliliters per day. That's the water gain. So to balance the water gain by osmosis requires the loss of about the same amount of water in the urine per day the salt and water in the feces is relatively small in this case. So it doesn't affect our balance very much. And the organism requires 240 micromoles of sodium per day. That active uptake is coming from the gills. And the urine output of 2.4 mils per day also takes with it 2.3 micromoles of sodium per day. It's a dilute urine. The concentration of sodium is 1 millimolar in the urine. And the net loss of 2.4 mils per day is 2 to 3 micromoles of sodium in the urine. So it's relatively small, around 1% of the loss of sodium is in the urine. The great majority is in the gills. Now, it's estimated that to balance the sodium and water in this organism has to spend about 3 to 7% of its resting metabolic rate just to handle the salt and water balance. Now, there are some exceptions that are interesting and give us important insight into these mechanisms. The freshwater crab, which occurs in Africa, Asia, and Europe, but they're not native in the Americas. I think they're being introduced in the West Coast. But we don't usually encounter freshwater crabs here, so we're not familiar with them. They tend to have a high blood osmolarity. See, it's around 5 to 600 milliosmoles, which is rather high. You remember that the ocean water is 1,000 milliosmoles, and freshwater organisms, terrestrial organisms too, tend to be around 300 milliosmoles or less. Now, they have UP ratios around 1, and so with that ratio, they lose sodium and chloride in urine. It's very high loss of those ions. And so what they have a compensation is that their carapace is extremely low permeability to water. And urine rates are kept very, very low then. You see 0.6 to 3.6 of their body weight, which, if you recall, is less than 10% of the other freshwater animals. Remember, they were 30% up to 100%. And there's a fish, the toadfish, that's very similar to crabs. And the idea is that these are relatively recent immigrants from saltwater to freshwater, and they therefore have a shorter evolutionary history and have not evolved yet kidney adaptations that would allow them to drop the blood osmolarity. The dilute urine here in these saves energy because less ion uptake then is required. So this is a type of adaptation, but not the adaptation that most freshwater animals have. And the thought is that they have not had time to evolve these more efficient mechanisms. Now, if we look to marine animals in the ocean, we have two examples to begin with, to contrast. One is the octopus. The octopus is a cephalopod, and it had the octopi have been in the water, in the marine water continuously. So they have a unbroken evolutionary history in salt water. And their blood tends to be isosmotic to seawater. So it's going to be between 900 and 1,000 or so milliosmoles. On the other hand, we have the teleos fish in B, and these fish in the ocean have come from ancestors that were living in fresh water. So remember the freshwater fish came from the ocean initially to freshwater, but all the teleost fish that are in the ocean now have come back from freshwater to saltwater. And so they tend to be hyposmotic because their ancestors were hyposmotic freshwater fish. Now, most marine invertebrates are isosmotic to seawater. And they don't have problems with osmotic regulation because they're living in an environment that they're isosmotic to. So they don't tend to gain or lose water or ions. Their blood tends to have very few organic compounds, mostly inorganic ions that are fairly similar to ocean water, some variations, but by and large they're fairly similar. Let's look at these in a little bit more detail. So these are the blood plasma or extracellular fluid concentrations in invertebrates and hagfish. Hagfish, very primitive vertebrate. And what you see here is very high concentrations of sodium compared to mammals, and very close to the seawater levels for sodium, potassium, calcium, magnesium, although there's some variations. There's a very low magnesium in the crab and in the hagfish. Chloride, fairly uniform. Sulfate's got some variations in it. This is seawater at the bottom. So the ion concentrations of these animals are very close to seawater. As I said, hagfish is a primitive vertebrate, and it's probably the only one that is doesn't have a freshwater or terrestrial journey in its evolutionary path. And it's the only vertebrate that has isosmotic blood and inorganic ions similar to seawater. So it's similar to the marine invertebrates, though this is an ancient vertebrate. The traditional evolutionary dogma here is that vertebrates began in the ocean as jawless fish, migrated to freshwater, developed jaws, and then migrated back to the ocean. This is what I was saying before. Originally, everything comes from the ocean. They go to freshwater where they get jaws, and then some of them come back to the salt water, back to the ocean. So their blood resembles freshwater invertebrates. So all jawed vertebrates and lampreys are descended from freshwater ancestors. There's a little bit more about this in box 28.2, if you can check it out if you're interested. So, whereas the freshwater fish were the hyperosmotic regulators, these are hyposmotic regulators. Marine teleost fish are hyposmotic, low osmotic regulators. Their blood in the range of 300 to 500 milliosmoles tends to be higher than the freshwater teleosts for the ocean fish. But that still means the osmotic difference is twice that of freshwater fish. So while freshwater fish, you are dealing with an osmotic difference of 300 milliosmoles, which is in their plasma, compared to 0 in the fresh water, a difference of 300 milliosmoles. The marine fish have a difference of 600 milliosmoles because they tend to be around, their blood is a little bit more osmolar than freshwater fish at 400, but the difference between 400 and 1,000, which is in the seawater, is 600. So they have a double the osmotic pressure difference, and it's in the opposite direction. And what this means is the osmotic pressure in the marine fish tends to cause water loss So water leaves the fish dehydrate, and they have to make that up by drinking seawater. But now you remember we said that it's difficult to gain fresh water by drinking seawater, but what these animals do is they have a place, an organ area where they can remove salt out of the gut, and that causes then a reverse osmosis, and so they can extract the fresh water, pure water, separate, at the cost of accumulating a large amount of salt. So again, volume regulation makes ionic regulation worse. And just like we talked about in the freshwater fish, but again, it's for opposite reasons. One of the ways they do this is that divalent ions, be magnesium sulfate, these are excreted in the urine of the fish. And they're relatively lower concentrations anyway. But the monovalent ions are excreted by gills. And so here again we have the separation between ionic transport and volume regulation, one being kidney, one being gills. Fish kidney can't produce a more concentrated urine than blood. They don't have UP ratios higher than 1. So the kidney can't help with osmotic regulation. It can handle volume regulation, but not osmotic regulation. And since water enters hyperosmotic into the fish and leaves isosmotic, that means you're going to retain solutes, which in this case is ions. And so the adaptation is urine rates kept very, very low. And again, these rates are in the range of 0.5 to 3.5% of body weights. And this is 1/10 or less than what you see in the freshwater fish that have, and freshwater animals that have very high urine rates, which is the opposite. Now, in the gills of the marine teleost fish, you have the same kind of salt excretion there in similar places in the gills that you had salt uptake in the freshwater fish. And there are mitochondria-rich cells, same type of cell, but these now are the marine type of mitochondria-rich cell compared to the freshwater type. And it's the picture here is given so that you get the idea that there's a lot of mitochondria in these cells because they're doing a lot of pumping. They have also a very interesting tubular system where there's huge invaginations of the extracellular or of the membrane, the cell membrane that invaginates. And they're all, it's an interconnected series of tubes that lead from the cell membrane, a huge surface area on these cells. And these are the cells that do the salt excretion and make the point that in marine teleost fish, the gills are the principal site of osmotic regulation because they can change the solutes in the blood, whereas the kidney in these fish cannot. Now, the method of salt secretion is these salt glands, which is a fairly common mechanism. There's variations in them, but it's a common mechanism that has evolved, and it works in birds and sea turtles, and also in the sharks, skates, and rays. So the cartilaginous fishes. So it's an relatively old evolutionary solution for pumping salt, for getting rid of salt. As you would expect for organisms that have originated in the ocean and being hyposmotic to their environment, where they have to do a lot of ion pumping. And here, and we've seen variations of this before, but this one, let's start with the ATP, sodium potassium ATPase, active transport of sodium out of the cell into the blood. And that sodium then is used here in this sodium potassium 2-chloride transporter, the NKCC transporter, which moves one potassium in, one sodium in, for 2 chlorides in. The chloride, as the chloride concentration increases in the cell, it goes out through the chloride channel. Very interesting channel. You'll run across this quite a few times in your studies and in your clinical years. And as that chloride leaves, sodium comes with it through the through the blood, from the blood out into the water, from the not just electrochemical gradient, but remember there's a electrical component to the electrochemical gradient that is driving the sodium out along with the chloride as the chloride leaves. The potassium is kept from accumulating in the cell by this potassium channel, which allows that potassium to leave. Now, we can just make a quick comparison here then between the water and salt relationships in a freshwater versus the marine fish. So just kind of clear this, make sure we have this straight. So in the freshwater fish, they gain water by osmosis, whereas the marine fish loses water by osmosis. They gain salt by diffusion in the gills, and the freshwater fish, on the other hand, loses salt by diffusion. They both lose salt and water in the feces, but remember this is a fairly small amount. The freshwater fish produces large amounts of urine that are very dilute to the plasma, and they have active uptake of sodium and chloride in the gills. They gain salt and water from their food. That is, they don't usually drink the water per se, but the water they get is from is what's in the food that they eat. That's where they get the salt. They get salt input from there as well. Now right. So this adds a little bit to the water load, but not a lot, and it helps with the salt uptake a little bit to supplement the active uptake. In the marine fish, on the other hand, they ingest salt and water in the food, and that's part of their net water gain by the water that comes in with their food. They, again, in the contrast, they have small amounts of urine, and it's isosmotic to plasma, rich in magnesium and sulfate, because those are the ones that are going to be excreted from the urine. The sodium and chloride are lost through the gills. Okay, so it's interesting because here you have the function of the gills and the function of the kidneys. It's similar organs, but they're doing an opposite function, taking up or getting rid of water versus taking up or getting rid of ions. And just the way we have a quantitative example for freshwater animals, here we have a quantitative example for marine animals, in this case marine teleost fish, and this example is given for a 1 kilogram flounder, for example. They have a water loss by osmosis that's around 8% of their body weight per day, and they are going to be drinking in equivalent of 11% of their body weight per day. And of that 11% of that they drink, they are keeping about 76% of it through that pure water uptake mechanism I mentioned earlier. In the salt and water loss that they have in the feces, again, is relatively small. About 2.7% body weight per day of the water loss comes out the feces. So it's a little bit bigger loss than you see in freshwater animals. But they produce, on the other hand, very small amounts of urine here, 0.4% of body weight per day. Nearly isosmotic to plasma, very rich in magnesium, so their UP ratios for magnesium and sulfate are over 100. And that's the way they get rid of magnesium and sulfate. The energy cost of this is somewhere between 8 and 17% of their resting metabolic rate. So the marine organisms have to use more of their energy to maintain their salt and water balance than the freshwater animals do. And again, it's about 2 to 1, which is very close to what the difference is between the osmotic gradients between the two types of animals. So that all makes sense. Here's some other things to think about in terms of the hyposmotic regulators. Many arthropods in the ocean and salt lakes are hyposmotic regulators. I didn't mention salt lakes as an environment. The ocean has about 1,000 milliosmol osmolarity, osmotic pressure, but there are salt lakes that have an even higher salinity than the oceans do, and there are animals that live in those salt lakes, and they are, like these arthropods, are hyposmotic regulators in the ocean. Sea turtles, sea snakes, penguins, gulls, whales, seals, other marine reptiles and mammals, got a wide variety of phyla, are hyposmotic to seawater and descend from terrestrial ancestors. So these are all phyla where the initial vertebrates, jawless vertebrates, came from the ocean, went to fresh water or the terrestrial habitats, developed, and then reinvaded the oceans, and so they tend to be hyposmotic to the ocean water. Blood tends to be around 400 milliosmoles, a little bit more than freshwater and terrestrial animals. Reptiles, birds, and mammals are air breathers. Now that helps them in a bit because they don't have their respiratory surfaces exposed to seawater. So remember, the gills in the fish are the site of oxygen transport, so they have a large surface area to handle oxygen, but that exposes them to the water, either the fresh water or the seawater, and it exacerbates the volume and salt issues. But in non-avian reptiles, birds, mammals, they breathe air, so their respiratory surfaces are not in direct contact to the water environment. And they also have very low permeability integuments, as we pointed out in the freshwater animals, they have developed tight integument, non-permeable integuments. When we get to the land animals, we're also going to mention that a problem when going to a terrestrial habitat is loss of water through the skin, and thus most terrestrial animals have an adaptation that makes their skin or whatever their integument is going to be very low permeability to water. So now when they come back into the ocean, they already have the advantage of having a very low permeability integument, and also they separate their respiratory. Now they do lose water from respiration in the air that's breathed out, and they also lose it through their skin, some through their skin. It's not a zero permeability. And they also gain salt from their food. Most of the animals in the ocean eat ocean animals that are salty, so they gain salt that way. Reptiles and birds can't produce a hyperosmotic urine, and so they need to excrete the excess salt that comes up. So, and thus they have salt glands. Now, the salt glands, the avian salt glands, are very specifically adapted to excrete high concentration of salt. And in birds, they are just above the eyes, in the orbit of birds, and they have a duct which leads out down the nose, either a duct that's partway out, like it is here in this fulmar bird, or at the end of the beak, as in the gull. And typical to secreting organs, they have a central canal, they're lobular, and the blood flow enhances surface area and presentation. And these salt glands are obviously very important to the marine birds, animals that live in the ocean. And they tend to be activated by osmoreceptors. The osmoreceptors may be in the aorta or in the heart, and they activate, their sensory input goes centrally, where then the outflow comes back out via parasympathetic nerves, and it's a acetylcholine system. So this is a neuroregulated excretion of salt. And this is important because the ability to excrete the concentrated salt means that they can extract pure water from seawater. And if you look at the concentrations of sodium in the exudate, you see it's very, very high. Remember, in the ocean, it was, here's standard seawater, 470 or so. We've seen around 500 millimolar for sodium concentration in seawater, and these are up to double the concentration in the by the salt gland. And it's in birds, turtles, and lizards. Now, if you look at, in this case now, we turn to sharks and the coelacanth. These are cartilaginous fish, the sharks, and they maintain an isosmotic or slightly hyperosmotic level, osmotic pressure in their blood. And you can see here the blood plasma osmotic pressure over 1,000, a urine just under 1,000. and the seawater that they're living in is 930. And you can see that high of a concentration of osmolarity is accomplished not by increasing inorganic ions, because you see sodium and chloride are kept unto what we would expect values of normal normal animals, regular animals. And the difference is that their osmotic pressure is made up by urea and TMAO. Now, the advantage of the urea is that they are excreting nitrogen using urea. And so this is a good way of getting rid of the nitrogen. But the urea concentration tends to, or allowing a high urea concentration in the plasma and in the cells, tends to interfere with proteins, causing kind of a denaturation of proteins, which messes up their function. And you remember a previous slide where we talked about pyruvate kinase. When the osmotic pressure goes up using these organic osmolytes, there's a compensating increase in TMAO, which tends to have the opposite effect on the protein, or actually be protective of the protein against the urea. So as long as TMAO is increasing with the urea, then you are maintaining the function of the ion. So this is an adaptation in these fish that allow them to have a high osmolarity in their blood without having high ionic concentrations, and they have a way of increasing their osmolarity by increasing organic osmolytes, and as they increase the urea that's needed to excrete nitrogen, they also increase the TMAO to offset the effects of urea on their proteins. It's a very neat balancing act that's being played here by these kind of ancient animals in the water. And the coelacanth is an interesting one as well. The coelacanth down here is doing the same thing. The coelacanth is interesting because it is on the ancestor to vertebrates pathway. So it is an ancient organism as well. It's usually found in great depths, and there's not too many of them found, studied. But it appears that the increased osmolarity of blood is an evolutionary old adaptation to living in seawater. And the original vertebrates are going to be coming from that type of an environment, as indicated by their ancestor, the coelacanth. And it doesn't change again until they go to fresh water and the terrestrial environment. All right, so here's a summary of the water-salt relations in a marine shark, cartilaginous fish. They have, again, they're going to gain salt by diffusion across the gills, and they're going to gain water by osmosis across the gills because they're hyperosmotic to the external, the ambient water, the ocean water, but they're hypoionic. Remember, they keep their ion concentrations low. So now they're gaining salt because they're hypoionic, but they're also gaining water because they're hyperosmotic. They get salt and water from their food that's preformed water. It's not that they drink it, it's the water that's in the food. And they are urinating modest amounts of modestly hypoosmotic that's rich in magnesium and sulfate. So excreting magnesium and sulfate in their urine, and they have a rectal gland, that's their salt gland, that gets rid of sodium chloride, and they also are losing salt and water in the feces. So the salt coming from the rectal salt gland and water from the feces is a way of getting water out. The exact roles of the gills in salt excretion are not really known very well. Now I mentioned briefly about the urea, and so getting rid of nitrogen is an issue that all organisms face. It comes, the metabolism of carbohydrates and lipids is much less of a problem because the waste products are water and CO2, and relatively easy to get rid of. But with protein metabolism, you tend to build up ammonia, which is the usual excreted form of the nitrogen that's metabolized from proteins. And the problem with that is that you can't have high concentrations of it. But an adaptation that allows a very effective way of getting rid of nitrogen is through making products like urea. And the urea is thus as the major alternative end product, but the problem with the urea is it's not cheap, it's not free. It costs 4 to 5 ATP per molecule more than just ammonia. So ammonia can be excreted more energy cheaper, but it can't be in high concentration and in high amounts. In order to get rid of nitrogen effectively, you need another compound, a nitrogen compound, and urea is very useful, soluble in water, very good, but it costs more energy. And if we look back evolutionarily, the cycle of ornithine-urea cycle was there in the very earliest vertebrates. And it's not a surprise. Remember, we talked about the earliest vertebrates descending from the jawless fishes and the coelacanth, which has hyperosmotic blood and urea and TMAO in the blood. So the function of urea was there very early on, but there's very much fewer vertebrates today that make use of the ornithine-urea cycle. So that has been lost through the movement to the new habitat, freshwater or terrestrial. Again, there's much more information on this in box 28.4 on the extension on the web. All right, let's take a little break at this point before we go on to the second half of the lecture. Take about a 10-minute break. All right, very good. So let's continue on now at the second half of today's lecture. And now we've talked about freshwater animals and their adaptations, and marine animals and their adaptations, and mentioned the fact that the freshwater animals have evolved from saltwater animals. And now it's really not too surprising to mention that there are animals that live in either saltwater or freshwater, or in partially salinated water. So animals are not limited to being in one habitat or another. They have preferential, but they also have existence outside that. So let's look at animals that now have to face differences in salinity. Now, there's always terms and definitions that we have to memorize and learn so that we can discuss them efficiently. First here, in terms of salinity, is a term called stenohaline. And the word steno, the part of the word steno means narrow. So whenever you see steno, it means narrow. Stenohaline would be narrow saline environment. And so stenohaline species can only survive in narrow range of ambient salinity. The opposite of that, species that can survive in a broader range of salinity, are the euryhaline species. Now, that's in response to the salinity. But remember, there's also osmoconformers. Osmoconformers, we talked from day one on the difference between conforming and regulating. So conformers permit their blood osmotic pressure to match the ambient osmotic pressure. So their osmotic pressure goes up and down with their environment. Sometimes the term poikilosmotic is used. This is like poikilothermic, again, the same roots and the same words. So if you see poikilosmotic, it's the same as a conformer. Osmoregulators, on the other hand, and again, analogously, we call them homeosmotic. They maintain a relatively constant blood osmotic pressure, whether or not the ambient pressure goes up or down. Most invertebrates in the open ocean are stenohaline osmoconformers. Okay, so they live in a narrow range of salinity, and they conform to osmotic pressure changes. Except oysters and mussels are euryhaline osmoconformers. So they can live in a wide variety of saline environments, habitats, even though they still conform. And in the term of osmoregulators, homeosmotic animals that regulate their osmotic pressure, these are of two major types. The hyperisosmotic regulator as one type, and the hyposmotic regulator is the second type. Now, the hyperisosmotic regulator, well, let's see what the differences are as we go along. So if we look at the A side of the graph here, the graph on the left, these are hyperosmotic, hyper-isosmotic regulators. Okay, they're all hyposmotic regulators, but the hyperisosmotic regulators are regulators when they're in lower osmotic pressure environments, but conformers when they are in a hyperosmotic environment. So what does that mean? That means that, let's look at the blue crab, for example, on this line here. So the blue crab maintains a fairly constant osmotic pressure in their blood, so they are a hyperosmotic regulator. They're regulating their blood to be hyperosmotic to the osmotic pressure of the environment. Until that pressure, until the external environmental water pressure reaches 1,000 milliosmoles. Now remember, that's the volume. That's the osmotic pressure of the ocean. So throughout the ocean, the blue crab tends to be a hyperosmotic regulator, although it gets less so as the osmotic pressure increases. And in osmotic pressures that are higher than ocean water, they become pure conformers. Here is another example, is in the amphipod, and they have the same feature where they are a regulator until they get to a higher value in the water and then become a conformer. And the carp down here, the carp is a regulator only in brackish type water that's below 300 milliosmoles. Above that, they are isosmotic. So this is the hyperosmotic or hyper-isosmotic regulators. Their blood stays hyperosmotic until the pressure gets high, and then it becomes isosmotic. The other type of regulator is the, in panel B here on the right, are the hyposmotic regulators. What this means is that when the osmotic pressure of the environmental water is low, they maintain a blood osmotic pressure higher than the environment, then they're hyperosmotic. But when the osmotic pressure of the water reaches a certain critical value, then as it goes further, they are hyposmotic regulators. So they maintain their regulation to very high environmental water pressures, despite the fact that they are now hyposmotic in this situation and hyperosmotic in this situation. There's a difference between the two. And you have examples of the fiddler crab in the blue and the ghost crab in the green, and brine shrimp. These are the sea monkeys that you may have seen in toy stores for feeding fish or for just watching them grow. And the shore crab. Now the shore crab, it's not that great of a regulator, but it's hyperosmotic as the osmotic pressure in the environment decreases. But it is poorly regulating here, but still it's less than, their blood osmotic pressure is less than the environmental. It's below the isosmotic line. So anything on this side of the line, anything on this side of the isosmotic line is a hypoosmotic regulator. And as you might imagine, the difference here is going to require different mechanisms. Now, to look at some further examples here of environmental switches, again, we have some terms. We have anadromous fish, and this is fish that lives in the ocean, but will go up rivers and streams to breed. And this is the classic example here is the salmon. Salmon lives its entire adult life in the ocean and then returns to the stream that it was born in, so to speak, hatched in order to breed. And when it enters a stream, it stops eating and uses whatever energy it has to get upstream and then still have enough energy to breed. That's its major challenge. That's anadromous. Live in the ocean and then breed up in fresh water. The opposite of that is catadromous. And these are fish that grow in fresh water, but they go out to the ocean to breed. They have to go breed in salt water. And the classic example of this is eels. And all of these two types are excellent euryhaline osmoregulators. Again, remember that means that they can survive in varied salinity environments, wide range of salinity, and they maintain their blood osmotic pressure fairly constant. Now, in freshwater fish, if you remove the hypophysis from the fish, and this is mostly the anterior hypophysis. So if you remove the hypothesis from the fish, they cannot do this adaptation back and forth. And the experiments described in the chapter will show you that its prolactin is required to reduce the permeability of the gills to sodium and augmenting urine flow. So switching to the freshwater model from the marine model requires reducing permeability and augmenting urine flow. Without prolactin, the fish can't do that, so they don't survive very well. They know this because if you take the hypothesis out, they can't survive. But if you add prolactin in, then they can survive. So the function here is the prolactin function. All right, now so here's an example of a study of adaptation, of acclimatization, or in this case, acclimation, in a study of trout that are acclimating to seawater or acclimating to freshwater. So if you take a freshwater trout, here's your trout, and trout's been living in freshwater, and at T0 here, you transfer them to seawater. And at various points in time, you measure the amounts of sodium potassium 2-chloride transporter, co-transporter, and sodium potassium ATPase in the gills of these fish. Then you find that over a period up to 2 months, they are increasing the abundance of both sodium potassium ATPase and NKCC, and the increase is 30 times in the case of the NKCC, 10 to 12 times for the sodium potassium ATPase. So there is an acclimation which is causing an increase, an upregulation of these transport proteins in order to acclimatize to the seawater. Now then, if you at some point transfer them back to freshwater, then over time they lose those proteins. So those genes are suppressed. And that means that exposure to salt is a trigger for a phenotype remodeling. Again, we mentioned this early in the course about phenotypes and genes and remodeling, and here's an example of a change in phenotype from a freshwater gill to a seawater gill. Now it turns out, and here's the pictures down here in part B of the figure in the lower half on the left. We have the seawater gill, and remember down here is where the NKCC is. You can see the red cells here are the ones that, the cells colored red are the ones that have the NKCC, and in the freshwater there are very much fewer cells that stain for the NKCC. Another interesting part of this now is that crustaceans have 3 times the expression changes in the genes, not gens, but genes for sodium potassium ATPase than teleost fishes do. So even more phenotypic or change in crustaceans than in the even in the fish. Now, in re-invoking chapter 1 again, with phenotypic and genotypic changes, the response to salinity changes by these animals that do it can occur in all of the 5 time frames that we talked about physiology changing. The acute responses, the chronic changes, which we just had an example of the chronic change in the trout, chronically exposed to seawater, change to salt water, change to seawater over periods of months. And evolutionary changes, that is, changes that occur over multiple generations, producing the speciations that we saw. But they also can occur in developmental changes and in the periodic changes as well. So the response to salinity can occur within all of the 5 physiological frames. There's examples of all of those. Another example of acclimation, a chronic change, is given in Figure 2813 in your book, and it's comparing the North Sea mussels that are living at a salinity of 30 grams per kilogram in the North Sea versus the lower salinity living Baltic Sea mussels that live at 15 grams, half the salinity in the Baltic Sea mussels. And as the salinity of the test water is changed, you switch the mussels one from the other. So it's a little bit of a complicated graph here. Let's see if we figure it out. So in here you have Baltic Sea mussels that are living in seawater that's ranging from about 2 to about 45 grams per kilogram, and this is the number that are working here at this level, surviving, working, pumping. So the range, the effective range is from here to here for the Baltic Sea mussels, where they are most surviving. And on the other hand, the North Sea mussels, they're living between 12 and 52. So there's an overlap in their population. So when you switch them, then over time, they switch. So now the dashed line here of the Baltic mussel looks a lot like the solid line of the North Sea mussel. And the solid line of the North Sea mussel looks a lot like the dashed line of the Baltic Sea mussel. So they represent their environments more than they maintain their species independence. The test here is ciliary activity, and the ciliary activity, that's how they eat. So the ability to have full activity, level 3 on the chart, is what gives you the sense of that's where they're surviving best. All right, so you have one change into the other, and obviously they change back. Now another type of adaptation is what happens to an animal that's in a water environment where the water environment evaporates, dries up. So this would be a dehydration challenge. And there are some animals that enhance their survival either from high salt or from low water. They allow themselves to dry. They become desiccated. They give up their water. This is called anhydrobiosis. Here's the phrase up here, or here it is here. Anhydrobiosis. Anhydro is not water. Bios is living, so this is not water living or desiccation. And this can be thought of as an adaptation in another way. In these rotifers, this rotifer population, if they are desiccated, they can outlive this particular fungus that attacks them. So in this particular study, you have fungus, you have rotifers exposed to fungus that kills them. So 100% of these rotifers exposed to the fungus are killed. If you desiccate on the day of exposure, if you desiccate them and wait a week and then rehydrate them, 100% will still die. Same at 14 days. But at 3 weeks of desiccation, the rotifers still live, but the virus is not doing very good. And now 21% of the, only 21%, 1 in 5 are killed, 40%, 2 in 5 are killed by the fungus. And if you go out to 30 days, it's less than 10% survive. So this would be a way of a population of rotifers to survive exposure to a fungus by outweighting them, desiccating, becoming desiccated for a month. And the fungus doesn't survive as well. The rotifer does. This is a famous desiccator anhydrobiosis organism. It's called a water bear. You can see it's about half a centimeter, 3 or 4, it's about half a millimeter. So it's a very small organism. And this is not necessarily the driest form, but these animals can dry out completely for long periods of time. In fact, this is a scanning electron microscope image of the water bear, also called a tardigrade. And of course, the scanning electron microscope specimen is in a vacuum. So these animals can survive without air or water for long periods of time and then become reanimated when they're rehydrated. So it's a very interesting organism that kind of suspends life when it desiccates. Has zero metabolic consumption and just suspended animation. All right, that brings us now to animals on land, terrestrial animals. And so here's a few things that are useful to know about land animals. Repeat this information again, is that animal life originated and spent most of its evolutionary history in water, in fact, in ocean water. The earliest animals entered land most likely to escape predators, but evaporative losses of water from these animals posed a tremendous threat. And so it continues then and gives us illustrates the fact that problems of water balance are the most pressing challenge for terrestrial animals. And we divide the animals up, some more definitions, to humidic animals. These are animals that whose habitats are restricted to humid and water-rich environments, as opposed to xeric animals, Xero, zero, it means dry. So xeric animals are capable of living in dry, water-poor microenvironments. And in all these environments, lowering integumentary permeability to water is key. Animals have to try to reduce evaporative water loss when they're on the land environment. And land animals lose water continually through evaporative losses, which includes integument losses and also respiratory losses of water, and also through urine. Urine is required, water is required for urination. So The water losses are integument, respiratory, and urine. The greatest one being the evaporative losses. Now, the permeability barrier for land animals is not necessarily what you see there. It's not related to the thickness of the skin or the number of scales of a lizard or a tortoise or things like that. The permeability barrier in mammals, birds, and reptiles is limited to a layer only about 10 microns thick, and it's a lamellar or layered structure that involves keratin and lipids. And this is usually in the outermost layer of the epidermis, a layer called the stratum corneum, or horny layer. And it's that's extremely important. And the fact that it's keratin mixed with lipids, the lipids can vary species by species, phylum by phylum. So there's a lot of variations based upon whichever are the particular lipids that make up their part of the barrier. And because these lipids are integral to the barrier, they can be affected by temperature. As you know, if you just like butter will melt at a higher temperature, or wax congeal at a higher temperature, the permeability of the barrier can be affected by temperature. Now, so this figure is an example. 2815 here is an example of the that transition temperature effect, looking at rate of water loss in this particular grasshopper. And as the temperature hits about 40 degrees C, it's quite warm, so it's 100 degrees, above 100 degrees, then water loss begins. That's the indication that there's been a breakdown in the lipids in that barrier. Most insects will have a higher transition temperature, but it could be as low as 25 degrees C or as high as 55 degrees C in other insects. So if the temperature reaches a critical temperature, then the animal, the insect, is in danger of survival, of not surviving because of tremendous increase in water loss. All right, so now if we look at the evaporative losses through skin, we have the range given here. It's listed here as resistance. The higher this number, the lower the permeability. Okay. So you see that the frogs and toads have some of the lowest. So these are amphibians, and you see amphibian skin does not have as low a permeability. And this means that these frogs and toads have to live in a humid environment. These are humidic terrestrial animals, and they have to be in a more humid environment. Then as you see, snakes increasing, lizards, birds, people have reasonable resistance to loss of water. And there are differences in within one species in different population. So in this graph, we are looking at the rate of evaporative water loss, okay, and versus body weight, because as we see, the water loss is a function of metabolic rate. And here we have two populations, a southern population and a northern population. And the southern population has a lower evaporative water loss for any body weight than comparable northern population birds do. So this means that within the same species there can be differences in the skin or integument permeability. So it's an environmental, it's a phenotypic response to environmental stimuli. Okay. Now, if we look at this group of animals, gives us another idea. Here we have ambient air temperature on the x-axis and the temperature of exhaled air on the y-axis. We're pointing out the respiratory loss. So we talked about the integumentary or skin losses of water. What's the relationship of the respiratory losses? The respiratory losses occur, if you remember from the last time we talked about the amount of water that air can hold is dependent on the temperature. And the higher the temperature, the more air, the more water air can hold. So as the air comes in at the environmental temperature, and it's warmed up to body temperature, it is gaining water in that air, and then if it's expelled at that temperature, there's a water loss. One of the adaptations that occurs is to decrease the temperature of the exhaled air. And this can be done by a countercurrent method where the incoming air, as it's warmed, is cooling the outgoing air. That's the countercurrent. So some of the heat in the outgoing air is taken away to warm the incoming air. And that can save some water loss. And if we look at these animals here, we see the dog, for example, in the blue line, has fairly high temperature of exhaled air, so that not a lot of cooling is done there. The wren bird in the red line does better than the dog. And if we look at these small mammals, they do the best. The isothermal line would be the air coming in and the air going out is the same temperature. So the small mammals, which would be the most sensitive to losses of water in their exhaled air, they have a mechanism that allows cooling of the expired air to save as much water as possible. Remember, the small animals have more problem with water loss because they have a greater surface area to volume ratio, being small, so relatively more skin per body. And also they have a higher metabolic rate, so they have more respirations per unit size than larger animals do. So this adaptation of cooling the exhaled air as close to ambient as possible is an evolutionary adaptation to water conservation. Now, I didn't point it out here. Yeah, it's on the other graph. On this graph, we have weight-specific total rate of evaporative water loss as a percentage of body weight. So this is mils per 100 gram per day. or percent of body weight per day. And you see, the smaller the animal, the higher the relative loss of water. Again, this is pretty much what I was saying before, is that the metabolic rate goes up. We know this allometric scaling. And so the respiratory rate goes up. And so the loss of weight-specific total evaporative loss is greater for the smaller animals. Now, they do have they have a higher metabolic rate, so their metabolic water production is higher. This ameliorates it somewhat. They're making a little bit more water, but they're also losing a lot more on total evaporative water loss. The larger the animal, the less the water loss, because the metabolic rate is less and because the surface area to volume ratio is less. There's a huge variation in the amount of evaporative water loss among different types of vertebrates. If we look down here at the bottom, near in the vertebrates, for example, these are all approximately in the same weight range, and their weight-specific total weight of evaporative loss, body weight per hour. Here's a comparison of the lab mouse versus the kangaroo rat. We'll return to this again. We've seen this already. Living in a dry environment, water loss being much more better controlled, but not as good as lizards. Very little water loss on the lizards. They have a lower metabolic rate, and also their exhaled air is the same temperature as the inhaled air. So there's no additional water loss there. Frogs and toads have a huge water loss that they have to deal with. They have to gain water during the day somewhere. Birds, intermediate, big difference between two different types of birds. Now, if we look at insects, now these are all very small except for the ghost crab. The ghost crab, if you look at the 44 grams, ghost crab is a little bit, approximately the same range as the vertebrates were, but the ghost crab has much greater water loss than the equivalent vertebrates. The worst of these bugs here, I don't really know what kind of bugs these are, but these small animals have a huge water loss and require a huge replenishment of water. These insects, even though they're very small and are bugs, they, compared to the ones just above them, have a much better control of water loss. Now, the other way water is lost in the terrestrial animals is through excretion of water in urine. So, controlling the water content of urine becomes another crucial tool in the toolbox for water control. And if we look at the ability to concentrate urine, the more you can more an animal can concentrate urine, the less relative water loss it's going to have. So if we look at the scaling of urine concentration, and these are all going to be mammals. Mammals are the best urine concentrators of all the animals. They're really the only ones that can really concentrate urine. And that ability is a function of body weight. The smaller the animal, the better the ability to concentrate urine. Again, the threat of water loss is greater in small animals than large animals. So this ability is a crucial feature in protecting smaller animals from water loss. See, there's a couple of ones that vary here. The muskrat and the beaver, they live in a water environment. So they have more access to environmental water. And the dromedary camel, the dik-dik, and the kangaroo rat, the Australian hopping mouse, these four animals live in a desert. You can read about them in chapter 30 when we get to it, and how they handle water loss. But they have a much higher ability to concentrate urine than their body size alone would predict. So you have environmental as well as metabolic drive to the phenotype of osmotic pressure concentration in urine. Now, if we again look at the allometric relationship between water lost or gained per day, this is the turnover rate. So water is lost and has to be made back up again. And so that water turnover in terms of milliliters per gram is here given for reptiles, non-avian reptiles. And there's a scaling from a 10 kilogram reptile down to a small one. And there's water loss, water turnover, but you see it's an order of magnitude less. This is a log-log scale. So this is an order of magnitude less than eutherian mammals, birds, or marsupials. The water loss by birds and marsupials higher than eutherian mammals, and much higher than reptiles and birds. So here are the numbers. The graph also gives you some numbers for the non-avian reptile here at the 100 gram equivalent is producing about 1.8 milliliters of water per day, whereas the eutherian mammal is producing about 14 milliliters per day, and the birds are losing 33 milliliters per day. Now, for some specific examples of adaptations, as we get to the last part of the chapter here, these frogs, so arboreal frogs, so these are frogs that live in trees, tree frogs, they have an adaptation that allows them to live in a slightly more drier environment. They have glands on their back, and they reach up and grab the lipid secretion and wipe it down across their nose and onto their chest. And they keep their skin covered with this lipid, and it acts very similar to the permeability barrier that we talked about before. The problems that amphibians have when they're in the land environment is they have inherently a low integument resistance to water loss. Remember, they had that very low resistance value. They also excrete their nitrogen through urea. Remember, urea is water soluble, so to get rid of nitrogen through urea, you have to produce water. You have to lose water. And their kidneys don't allow them to concentrate their urine more than their blood is. So this puts a tremendous strain on water conservation. So another example we have is from the insect world, and these are ants that live in the desert, and they have long stilt-like legs, and these legs mean that they are higher off the sand. The higher they can get off the sand, the cooler the air is. There's a tremendous heat gradient coming out of the desert sands. And so This adaptation allows them to walk around on the sand for longer periods of time. And these ants live on ants that have shorter legs and that dry out and die in the desert. And then these ants go out and grab them before they're completely baked and dried out, and they get water from eating those dead ants. So that's their adaptation. Now here's another example of a change in adaptation over generations. So there's generational responses to an environmental change. And there's an interesting set of experiments that was done by putting flies, and this can be found in box 28.6 if you're interested in all the greater details. But in drier and drier environments, remember insects can suffer from dry environments from water loss as well. They're small, so their surface area to volume ratio is very, very low. So in this particular study, flies were put in a dry environment until 80% of those flies died. And then they took the 20% survivors, and then they made them the parents of the next generation, and they kept drying the environment more and more and more until they were selecting those animals that could survive dryness more. So as the number of generations of selection increased up to 37 or so, I forget how many they did, relative to the control flies exposed to dry air, their survival rate increases so that by the 37th generation, they can survive in the dry environment 30 hours more than the control animals can. And these flies tend to be a little smaller, but they tend to have more water per fly. So they are conserving water, and by 37 generations, again, they have added another 0.2 microliters of water per fly. So these are phenotypical changes for these generations adapting genetically to the dry environment. One of the classic examples is the kangaroo rat. This produced a tremendous amount of studies by physiologists to understand kidney function and water balance. And we've already run across this guy previously when we compared their ability to get water from their food. They don't need to drink in their dry environment. They can get the water directly from food. Let's look a little bit more into this guy. So in figure 28-24, this is the summary of the story. So this is a study, 25 degrees centigrade. These are rats that are fed air-dried, husked barley grain, but no drinking water. You remember that the barley grain contains an amount of preformed water, so they can get water from what they eat. So that's this. So their specializations are that they have an exceptionally low cutaneous permeability to water. We saw an example, it's one of the dots on the graph before. They also cooled their exhaled air via countercurrent exchange as much as possible. Remember, the small mammals can cool their exhaled air down to ambient. They can concentrate their urine. They have a UP ratio of 14, which is pretty concentrated for the osmotic pressure of the urine. And they have practically no or zero fecal water loss. The amount of water that goes out in the feces is less than the water that came in with the feces, so with food. So they don't lose any water through their feces. Let's look at the graph in a little bit more detail. So what is shown here is that the loss to evaporation, water loss to evaporation, okay, is this line here, and it scales with the humidity. So the drier the air, the more water that's lost. This makes sense because the rate of water loss is equal to the difference between the water saturation in the skin versus in the environment. So that scales. If we add onto that the loss to urine, which is a constant, so the distance between the distance between these two lines is constant the whole way up. It's not temperature dependent. That's the loss to urine. And the loss to feces, which is very, very insignificant, also independent. So the total water loss is humidity dependent because the evaporative loss is related to temperature. Now, if we look at the metabolic water that's being produced, this is the H2O that comes from oxidative metabolism. That's here, this thin blue line. That thin blue line is more or less constant across the humidity. The metabolic rate does not change. The water that they gain from food, and this is even though it's dry food, it still has water molecules trapped in it. H2O, preformed water, and that is humidity dependent, and it's greater. So the greater the external humidity, the more water is in the food. Okay? So anytime the thick blue line is equal to or above the thick red line, then their total water intake of these kangaroo rats is greater than their total water loss, and they don't have to drink anything. They don't have to add water. And it's only in the very dry and severely dry humidity environment that they need the water. Now this is all done at 25 degrees C. If you increase the temperature to 30 degrees, then this becomes more of a problem because the water loss is going to be enhanced by the temperature difference. So while this works well at 25%, but 25% is about where they're living in the desert anyway. They have to keep in a cooler environment to keep their water loss from exceeding their water intake. And the way this is usually expressed, because their metabolic water production is the major, let's go back here and look at this. The metabolic water production is the greater percent of the total water available, and the evaporative water loss is the greater of the water loss, of the loss to evaporation. One way to look at this is to look at the ratio of metabolic water produced to evaporative water loss. When this is above 1, there's a net increase in water by metabolism compared to what's lost by evaporation. And all these, and it's easier to maintain that at the lower temperatures. Okay, so here's even a bird here. It's fine up to 21 degrees. Less severe desert rat, the more severe desert, this is the one we were looking at before. They can maintain, but if the temperature goes above 30, then the metabolic water production falls below the evaporative water loss. And so the rats can't maintain water balance by just eating the dried grain, but have to supplement the diet with insects and green plant parts. As far as being hyperthermia tolerant, birds tend to be fairly hyperthermia tolerant. Mainly, one of their advantages is they have a higher body temperature. Their body temperature is 39 degrees compared to mammals 37 degrees. Also, they can fly to a place where there's water, so they don't have a limited range. If water's within their range, they can fly to it at a longer range. And they excrete uric acid. It's insoluble, so they don't have to lose water to excrete the uric acid. And so they just, in general, the design of birds, the overall design makes them more favorable to desert life. But there must be more, because birds that live in desert-type environments do show other adaptations that are specific. They have lower evaporative water losses. They have lower metabolic rates and decreased activity of life to go along with the metabolic rate, and smaller clutches of children, so baby birds. So in the case of as water becomes less available, so this is the index of water availability, and so this environment. So down here you have the desert environment, and up here you have a less arid environment. And in arid environments, the rate of water loss is less. So evaporative water loss is less in the drier environments in these birds. Field water turnover, so remember this is the loss and gain per day, is also less as a function of decreased activity, really. Basal metabolic rates are less in the dry environment. Activity is less in the environment. All of these, and smaller clutches per breeding season. All of this allows water conservation. So most of these birds aren't flying away to bodies of water to drink, but remaining in their existing locations, and have these other adaptations to allow them to do that. So the control of water and salt balance in terrestrial animals is mostly under hormonal control. We didn't talk about that yet. We did mention a neuronal control when we talked about the avian salt gland. There's a neuronal contribution. And so now we talk about diuresis, which is the production of abundant, usually dilute urine. And this is promoted by diuretic hormones. And they're the opposite. Antidiuretic hormones slow down diuresis, slow down urine flow. So this preserves water at the expense of concentrating the urine. Vertebrates use only antidiuretic hormones. So the control of volume in humans, in vertebrates, is only through the presence or absence of an antidiuretic signal. Insects, on the other hand, have both a diuretic hormone and an antidiuretic hormone. So the diuretic hormone, the example that the book gives, is in, for example, insects or mosquitoes that have a blood meal, triggering a diuretic hormone that gets rid of most of the fluid gain from the blood meal, to concentrate the particulate part of the blood meal, to shed the water. And there are three important hormones in vertebrates that are there to study. Antidiuretic hormone is an obvious one, made by the neurohypophysis. And this controls pure water excretion. So when antidiuretic hormone goes up, H2O excretion goes down. Then you have the mineralocorticoids, and the most famous one here is aldosterone. This comes from the adrenal cortex, or whatever homologous organ in the vertebrate does the job of the adrenal cortex. And the action of this hormone is to retain sodium, but it promote excretion of potassium. So it increases the sodium-potassium ratio. And then the third are the natriuretic hormones, which promote sodium excretion. The natri part of the word is from sodium. Natrium is sodium. And uretic is urine. So natriuretic means sodium in the urine. That's sodium excretion. It's a way to remember that this promotes sodium excretion. And that pretty much does it for today. I know it's a long lecture, a lot of stuff involved in that, but it's really putting together a lot of the concepts that we discussed in earlier chapters. Next we have the last of the regular chapters will be the next lecture, chapter 29, and then the final lecture will be on the life at work. We'll look at animals that have adapted to the desert life and how that goes. All right, so I'll see you next time.